Thanks for having me here, and it's an honor to present to this audience. First, the usual disclosures. If we can get to the first slide here. Let's go back. Let's try it again. All right, no disclosures. Let's go. Don't know what that means for a hospital-based system here. All right, so I have a lot of things to disclose. You probably shouldn't believe another word I say over the next 15 minutes. Start a bunch of companies, consult for a bunch of companies. What I'm really excited about is technology that you're seeing on the slide here. Let's just get right into it. So I'm really here to talk about big data and biomedicine. I think we just had a panel on big data, looking at it from the healthcare perspective, from the systems perspective. And here I'm going to get a little bit more molecular for a reason. I think a lot of excitement in the future of molecular diagnostics and the practice of molecular medicine. And it's kind of an interesting coincidence that this meeting is actually just right across the way from the American Society for Human Genetics, another meeting I'm attending right after this one. It's kind of neat that the two future directions of medicine are right next door to each other, the social connected side and the genetic side. All in one place, of course, has to be in Boston. What I'm showing you on the left here is just a nifty device. It's just one example of a high throughput technology in life science research and biomedicine. And I have one in my hands here if you actually want to see what this actually physically looks like. It helps to have a prop sometimes because big data seems to be virtual. Here's a physical chip. And this looks like a computer chip by design. And you can put any sample on this, let's say a sample of cancer or diabetes or healthy tissue, and gets a readout of all the genes in the genome. How much is it turned on or off? What level is it made at? And then when you get tired of running these one at a time, you can run them 96 at a time. Some of you might spend time in a lab, you know, these 96 well plates. Well, now you have each one of these chips is one well here. This is a nifty technology. It's just one of many that we have in high throughput biology today. But what's truly amazing is that this is already a 15-year-old technology. We've actually had this for 15 years already. And every major academic medical center, every university runs these chips today, and many other types of technologies. And what happened in the early 2000s is that everyone started writing papers about these at Stanford and Harvard and everywhere else. And then the journals were the first to say, the journals said, hold on a second, we're getting too many people, too many researchers using these types of tools. No more papers with these chips, no more papers, unless you put this data into an international repository. So other people can kind of double check your math. It's important to double check the math. We're getting closer to the clinic, and maybe use these samples for something else someday. And what I'm here to tell you is that we're in this magic world where we make scientists share their data on the internet. What an amazing concept. And as of, we hit a milestone last year. Nature had this special article. The milestone we hit last August 2012 is that we hit a million of those chips publicly available now. 1 million samples. Actually, that was last year. If you go to the websites today, these are the two screenshots of the two websites, one at the National Library of Medicine, one at the European Bioinformatics Institute. We're already at 1.25 million, and we've already added another 250,000. We're finally slowing down to Moore's Law. Slowing down to Moore's Law, okay? Moore's Law's computational power doubles every 18 months or so. We're no longer quadrupling or tripling every year. We're slowing down because other repositories are growing. And just to kind of make it amazingly obvious why this is the niftiest thing ever, right? In the middle of this website here I'm showing you, it's just a simple search box. And if you just need to search for samples, or, you know, like I say all the time on my talks, imagine a high school kid now. Forget about a researcher or Mass General. A high school kid that needs to do a science fair project, right? She can type in breast cancer here on this website and click go. And now she can find and download 39,000 samples of breast cancer for her science fair project, about as easily as she could find a song on iTunes, right? Imagine a science fair kid doing a project now, starting with nearly 40,000 samples. And you know what, if that's not enough for you, in 2 years it'll be double that number. Now that's an interesting concept, 40,000 samples of breast cancer or colon cancer, or prostate cancer, whatever disease you want. Some researcher somewhere has studied it and has already had to share it on the internet, these molecular measurements. The number I like better is 1,500. That's a number right above that. 1,500 labs have already generated some data making up this set of samples, 1,500 labs all across the internet. Let me tell you how amazing that is. Let's say I was going to start a brand new effort on solving breast cancer. And I'm going to get my friends on the internet to do this with me. So kids today call that crowdsourcing, right? right? So I'm going to crowdsource my breast cancer. And I'm going to get 1,500 of the best labs in the world to take their best patients, to run their best measurements, and share all that data with me for free. If I proposed this in a forward direction, you would laugh me out of the room. Why would any one of those 1,500 share data with you for free? But here we are, that in some ways, what I call public big data is retroactive crowdsourcing, right? The crowd is helping you, and they don't know it. And the crowd is helping you, and you don't actually even know it, right? But here it is. The data is there, and it's all available for free, and still doubling. And you don't have to do anything about it to get it. So this retroactive crowdsourcing, to me, is the magic here, is that you have these experts running these samples. They're waiting for you to do something amazing with them. And so if you have any doubts a high school kid can do this, here is a high school kid who has done it. So I've never met this girl, Brittany Winger from Sarasota, Florida, 2 years ago, won the top Google Science Fair competition because she came up with an artificial neural network to diagnose breast cancer using publicly available data. And if that wasn't enough at age 17 last year, or earlier this year, she did the same thing again now for leukemia, and she was on the front page of major newspapers. You know, Jack Andraka is another kid. He was in the 60 Minutes last week. Kids can do amazing things. And of course, what I'm saying is if a high school kid can do it, of course, we in this audience and other audiences should be able to do this too. It's just sitting there waiting for researchers to use this data. And so my first big grant at Stanford from NIH was just go get every human disease out there. Just go get them all. We've got 1 million of these chips. Just go get every one of them. And we build big tables like this, where the rows are all these different diseases like leprosy and neurofibromatosis and of course cancers and asthma. And all the columns are all different genes that are being measured. And we know, does the gene go up or down, and how much does it go up or down? And then we start to see interesting patterns. This disease and this disease look similar to each other, but they're completely different kinds of doctors that take care of these diseases. For example, muscular dystrophy shows up next to having a heart attack. Well, you got pediatric neurologists on one side, adult cardiologists on the other, and these two docs hardly ever even see each other in the hospital system, but their diseases are very similar to each other. And that got us to thinking, what can we do with that kind of information? And along came other groups, like the Broad Institute, Whitehead, and many others now, that started to study drugs this way. Take cells, cancer cells, other cells, with drug, without drug, with drug, without drug. So on the left, I've got patients with disease, without disease, with disease, without disease. And on the right, with drug, without drug. And I'm going to make the statistics really simple here, okay? I'm going to use my arms. If I've got a disease now that makes this gene go up and this gene go down, and I find a drug on the other side that makes this gene go down and this gene go up, that might be a partnership there, right? The reporters call this Match.com for drugs. Why can't I get the drug matched to the disease here, right? Especially if, first of all, if that drug is really starving, looking for a new use, and especially if that disease has no other therapies. And so we start to play this game. Of course, you can't be naive. Not every drug hits every tissue in the body, but we started to use this public data to make these kinds of marriages kind of made in heaven. And our first best use of these are my famous rat colonoscopies. Think about those two words together and you realize why this is funny, rat colonoscopy. And here we showed that a seizure drug, topiramate, actually seems to work on Crohn's disease, inflammatory bowel disease. So on the left, you'll see a normal rat colonoscopy. In the middle is the rat with this Crohn's disease, given a chemical to make this happen. And on the right is what happens after it's treated with topiramate. And we got really excited about this. And then we've done a bunch of these. I've lost count at this point. My favorite one is this one. It showed up 2 weeks ago. It was in some of the newspapers you might have seen. This is a mouse that gets lung cancer, small cell lung cancer, neuroendocrine tumor. And so the rest of us have to smoke to get lung cancer. This mouse has these 3 genes knocked out. It gets its own lung cancer. You see some nodules there. And on the right is the same kind of mouse treated with an antidepressant, okay? Tricyclic antidepressant. We don't use these anymore. They have some annoying side effects, arrhythmia and stuff like that. Nothing as bad as having lung cancer, though. And the mouse on the right, you can see the cancer is melted away. It's gone. And so here's an antidepressant that's still in the pharmacy, which we really don't use, melting away small cell lung cancer. Now, why am I showing you this one? Because this is the idea of drug repositioning, finding new uses for drugs that maybe we could still do something more with. But we had a computer prediction. We got cell lines working in a dish. I'm not even going to show you that. We got this mouse that seems to be doing better. In 15 months, from computer 15 months later, we get the ethics, the IRB approval, and we got patients already recruited in a trial. And this, to me, is the next level, right? To go from public big data like this to getting drugs to patients in a trial. I don't know if this drug is going to work. I hope it does. I'm actually not even involved with the trial. But I think this is what we should be trying to do. It's not just playing with this data, not just getting annoyed we had to share this data, but coming up with new uses to get more drugs to patients. And with great collaborators working on every step of the way here. Of course, the Stanford way is once you get this to work, you got to spin out a company on this. And so we started a company. And NewMedi is an example of a company that's just taking this same platform and launching it. They just got their Series A couple months ago. And the idea here is we realized that it's important to pharmaceutical companies that are actually looking for something to sell. If you haven't heard, the pharma industry is going to have problems by 2017, 2018 in terms of not having enough to sell. Now, you might like the pharma industry, you might hate the pharmaceutical. I'm not going to pass judgment on them. But it's something wrong if they're going to lose $250 billion of revenue in 2018. That's a big deal. I worry about the future drug discovery if they lose that kind of revenue. So I think we need alternative approaches to come up with these kinds of drugs and ideas. So another source of big data, again, exactly relevant to the conference right across the street here with the genetics, is the Genome Project. So for those of you who don't know, the Genome Project was completed in 2003. And it's to get 6 billion letters. 6 billion letters, 3 billion come from your mom, 3 billion come from your dad, and they're in chromosomes. But essentially 6 billion letters all read out in some sample humans, and now we can do this on individuals. The Genome Project was completed in centers like the ones you see here. On this side, this is at the Whitehead Institute, just right across the river, just an example of robots picking bacterial clones and a staff of people running this. And literally where we're heading is towards doing all of this, not in 13 years, but to do this within hours on a USB stick, right? A sequencer, not in a room like that, but running in a USB stick. Now, there are a lot of companies proposing this. We'll see if these actually become real products. But it's a no-brainer this is going to come to medicine. It's a no-brainer. Already the price of getting a genome done is cheaper than a mammography or cheaper than a colonoscopy. I think it's a no-brainer. We're going to have to look at this. And so what we realize is that this is generating personal big data. 6 billion letters, it's a lot of books if you put them all into books. And we start to realize we need to use published big data to interpret this big data. So we started an effort in my lab to literally just go read every single paper in human disease genetics. You can barely see here. These are the front pages of 1,000 papers. We've actually read 20,000 papers now and just distilled the essence of all of these genetics papers and put them into computerized form so we can interpret these genomes, because we were getting more and more patients appearing, coming to our practices at Stanford with their genomes in hand. And so what we can do now is come up with what we call riskograms. And so here you can just see disease by disease. This is published in The Lancet. If you're really curious, you can look up this paper. But at the top, heading down, you see all the different diseases that someone can have. So obesity, type 2 diabetes, stroke, migraines. The black arrows indicate what we thought an individual's risk was for having this disease based on epidemiology and statistics. And the red lines indicate for that one individual the new risk that we have given that genome. And the blue lines indicate some protection. So this individual is less likely to get Alzheimer's in the population and is more likely to have obesity, coronary disease, and type 2 diabetes. You know, I'm not even concerned anymore about coming up with the exact fine-tuning of these percentages anymore. I think the value of the genome in some ways is if I could put you into one of 3 buckets, right? Are you going to have a problem? Look, we're all, not to be morbid, but we're all going to die here. As far as I heard, in Boston, they haven't found a cure for that yet. But if we're all going to die, if there's one of 3 buckets, whether it's going to be cardiovascular, cancer, or neurological, one of those 3 buckets, which one are you going to fall into? And it might be what your parents had or maybe not. And the prevention strategies, as we've been hearing a lot about at this meeting, the prevention strategies are radically different for each of those 3. So even if we could just put people into one of those 3, I think there might be some advantage here. But when I think about genome sequencing, I think about this other story. If you've never heard of this kid, Nicholas Volker, this work done out of Milwaukee, and this is a Pulitzer Prize-winning story. So I recommend after this meeting to go Google this guy up, this story. This is a kid who spent the first 5 years of his life in the intensive care unit. The first 5 years just getting bowel surgery after bowel surgery because he was losing his bowel. He was infarcting his bowel. And he's an N of 1. Nothing's ever been written before on a syndrome like this. So what do? Well, after 5 years of just repeated surgeries, all of a sudden they realize there's no hope left. let's get the genome sequence on this child and the parents. And all of a sudden, they see a new mutation in the child that neither parent had, in a gene that actually hangs out in the lymph nodes. And this gene gave them enough of a clue that they said, we should go try a stem cell transplant in this kid. Okay? So think about like reimbursement and payers and practice guidelines. Here's an N of 1 kid that has no history. We don't even know what could happen with this kid. They went ahead with this, and now this kid goes to school and eats a diet, a normal diet. Right? So this transplantation cured this kid. This is, in some ways, the first child ever saved by DNA sequencing. And this is literally the poster child for DNA sequencing, what we can do today. Never mind, in some ways, getting this entire human genome sequenced this way actually might even end decades or at least years of diagnostic odysseys where parents and children are looking for answers. What does my child have here? And along the way, collecting expensive tests that might come to an end once you get the entire genome this way. So, but big data is more than just molecules. I'd like to show this slide here. So this is at the molecular level. I mean, I show you at molecular level. This is more at the personal fitness level. So some of you might have Fitbits or Withings. I think there have been a couple talks on personal wellness. There's some displays over there. How many of you have a Fitbit? Let's just see a show of hands. Just about maybe 10%, and they're all sitting in the front. So it's kind of interesting here. Very interesting. Yeah, it's early adopters for sure. Okay, someone all the way in the back is raising their hand too. So whether you have Withings or Fitbit or Nike or Jawbone or any of these kinds of companies, these are devices that count your steps. You can get the scales. You know, I kind of used to laugh at these quantified self people, right? I mean, they count everything going in and out of their bodies until my weight gain was getting ridiculous. That's my scale at the bottom there, my BMI curve. I'm putting it out there. And you can see I was in the red zone until January, when I started to measure the scale. I have my Fitbit in my pocket here, just counting my steps. And now I'm in yellow. I have a long way to go until I get to green, but I've lost 35 pounds just since January doing this, right? So there is something to this, okay? There is something to this. So just track, you know, the age-old saying, if you don't measure it, you can't change it, right? I mean, everyone knows that in any space. So I think this is kind of an amazing concept. It's also about the charges and the costs, and we were talking about that a little bit earlier in the panel. I think I was at University of Miami giving grand rounds when this showed up in the Washington Post, that University of Miami charges double what the neighboring hospital charges, Jackson Memorial, right? I apologize to anyone from Miami here, but this is kind of painful. You want to know what your charges are and your costs, whether they're average or individual costs. It doesn't really matter. You want to know your big data before the reporters do, because this data is becoming public, and people are just going to have apps where they can spin around which one's cheaper and which one has better quality, and the apps are already there. There's even an app where you can look to see whether the hospital cleans the bathrooms every day, okay? Not kidding you, these are real apps now. And I think the next big data, what I'm most excited about now, moving forward, NIH gave me this big grant to start to think about clinical trials data. And clinical trials are super expensive around the world. More than half of them fail. They never make it into publications. We never learn from the science that's done from a failed trial. Imagine a world where we get raw clinical trials data, de-identified, of course, but what if we could learn in real time what these trials are showing? Right? Think about it now. We run an expensive trial, then we wait for the physician scientists to write a paper. 2 years later, it's published. Then we wait for a doctor to read the paper and then implement the practice. What if your EHR someday could say in the morning, oh, this is the 4th trial I've seen the raw data where this drug seems to work. Let's implement it today for patients, right? And just put up a flag and let's let the right committee review it today, and let's start implementing it today instead of this years of an arc. But I think it could be incredible apps built on top of just clinical trials. We've already released 60 clinical trials. I think it's the most so far, and you can get many more as the weeks go on. So it's just the beginning. There's so much data out there. There's genome projects and ENCODE and cancer cells and more drugs being studied. Of course, epic concern of the data we're capturing in electronic health records, and all sorts of countrywide sequencing projects, pharmacogenomics. This is just the beginning, and this is all publicly available data. The EHR data is not public yet, I grant you that, but in some ways we're going to get snippets of it through the trials in some ways. So in the end, what is big data in biomedicine, right? What is this concept? Is it about algorithms, programmers, databases, cloud computing, mobile, high-performance computers? I actually think big data in biomedicine is about none of these things. I think big data in biomedicine is about predicting the disease before it strikes, explaining the rare disease that defies our experts, finding drugs for diseases that lack attention today, making sure we do the right, safe things for patients. And it's an amazing platform for biomedical innovation and entrepreneurship. And I can sum all this up with one word: big data and biomedicine is all about hope. For patients, for families, for providers. I think that's what's the most amazing. It's not the technical stuff. It's about us really changing the world of medicine, because we're all going to be able to get to this data and just make amazing innovations and discoveries from it. So I'm going to put in my own plug for my own meeting, Big Data and Biomedicine, in May. I can't do any of this work without a long list of collaborators. I have to thank the Packard Foundation for funding me, as well as the NIH and many other disease foundations. So thank you very much.
Absolutely awesome.
Thank you very much.